Hello everyone. So the next topic which we will be going to study is about the mass flow hypothesis. So let's start with the definition first, that what is mass flow hypothesis? It is the movement of substances in the bulk from one point to another as a result of pressure difference between the 2 points. So this is the definition of the mass flow. So there is a long distance transport of substances which is within the plants, and they cannot be done by the process of diffusion alone. So there is a special long distance transport which is also called as translocation, in which the substances move across the long distance and at a much faster rate. So this process is carried out by the mass flow. So this is known as the mass flow. Then what is mass flow hypothesis? What is the history of mass flow hypothesis? So this theory was proposed in 1930 by the German plant physiologist, that is Ernest Munch. What he stated, that the translocation of glucose and sugars within the phloem, it is caused by a never-ending flow of water and dissolved nutrients between the sources and the sink where the sugars are basically utilized. So through this hypothesis he concluded the process of translocation and in the phloem. So a diffusion gradient or osmotic gradient is caused by the phloem sources cell due to the high concentration of sugar and other organic substances. This results in water being drawn out from the adjacent xylem, and there is a hydrostatic pressure which moves the sap. So this theory is very closest to the explanation available to describe the mass flow in the phloem. So we are mainly focusing on the translocation process in the phloem, or the mass flow in the phloem. So how this works, and what is the mechanism for this mass flow hypothesis? So as we have discussed in the previous slide also, that this is a translocation process in which the movement of minerals and water through the xylem, which creates a negative pressure, and they move in through the phloem as a result of the hydrostatic pressure. So this movement is accompanied by the process of phloem loading and unloading. So this is the process by phloem loading and unloading. Now mass flow hypothesis. When the plants respire through the process of photosynthesis, it involves the formation of glucose in the mesophyll cells. So in this, all the sugars are not utilized. Leftover glucose becomes non-reducing sugar. So when the glucose is left out, it will become a non-reducing sugar. So other examples of the sugars are sucrose, which is delivered to the neighbor cells, and that is present in the veins of the leaves. So if we talk about the plasmodesmata, through the plasmodesmata, the sugar diffused from the neighbor cells and they enter the sieve tube. Sieve tube is a component of the phloem. So it will enter the sieve tube, and the number of sugar within the sieve tube and its elements that will increase. So this is the beginning of the mass flow hypothesis, which is mentioned here. So this is how a mass flow is being begin, or it will be initiated. So at the same time, when the phloem is transporting the food, at the same time the water which travels from the nearby tissue through the xylem to the source cells via osmosis, they create a increased hydrostatic pressure in the elements of the sieve tube. So this is the mass flow hypothesis for the mechanism of translocation in the plants. Now the hydrostatic pressure, basically what happens is, the hydrostatic pressure, it will shift the sugar and the substances down the cells of the sieve tube towards the sink, that is the roots, towards the sink. So when at the root or at the sink level, the sugars, they are destroyed and they become apoplast, and then they enter the symplast. the water that then will travel out of the cells and they reduce the hydrostatic pressure in them. The gradient which is created is the result of the pressure developed by the entry of the sugar and the source and the elimination of it at the sink. So the learning of mass flow hypothesis is at a level is more complex. So this is what happens in the phloem at the tissue level, at the translocation level, where the mass flow hypothesis begins. So what happens next is the root cortex and stem, they eliminate, and the phloem sugar uses it for the cellular respiration. So starch does not cause any pressure as it is insoluble. So at the end, through the transcription pull, the pure water left is absorbed. Now this is a diagrammatic representation of how the flow of mass flow hypothesis is being depicted, and it is a 6-step process in which whole of the mass flow hypothesis is being depicted here. So step number 1, this is a sucrose, so this is a source leaf, and inside is the sucrose. So there is a water absorption from nearby cells which is increasing the hydrostatic pressure in the mesophyll cells. So there is a increasing hydrostatic pressure. Just near the source there are companion cells. What is companion cell? Where the phloem is been loading. So phloem is been loading here. There is a transfer of organic food from mesophyll cells to the sieve tube. So this is the sieve tube elements also, and this is the companion cell. So when the sucrose is being produced in the plant or in the leaf cell, it will then be transferred to the companion cells or to the sieve tube. So this is the process of phloem loading, when the food is being transferred to the companion cells, the sieve tubes. Then what happens is water entry from the xylem to sieve tubes by the osmosis. So water is also entering to the xylem to sieve tubes element by the process of osmosis. So the black arrow here is representing the 3rd step here. Next, the 4th step is there is a hydrostatic pressure difference which drives the solute movement from sieve tubes to sink. So there is a hydrostatic pressure which will create or which will drive the solutes movement from the sieve tubes. Next, what happens is step number 5, the phloem unloading. Here in the step 2 there was phloem loading, and in the 5th step there is a phloem unloading, in which what is happening, the transfer of organic food from the sieve to the sieve tube to the sink. So this is the root cell. Sink here is representing the roots only. So what is happening, the red arrow, as you have seen, sucrose has been transferred to the sieve tubes in the phloem, then it will be transferred here, it will be transferred to the companion cells, and then again to the sink, that is the root cell. So this is the phloem unloading, when the food is being transferred from the sieve tube to the sink. This process is done. Then the last step, the 6th step, what is happening is water moves back to the xylem vessels. Here the water was moving towards the sieve tubes because of by the process of the osmosis, but in the last, when there is an unloading of phloem which is being done, after that what is happening, the water moves back to the xylem vessels by the osmosis, and transpiration pulls up the water. So the transpiration will pull up the water here. So this is the whole diagrammatic representation of how this process of mass flow hypothesis works in the 6 steps, through xylem, phloem, through phloem loading and phloem unloading. So it was all being described here. So these are some of the references.